Hey, are you interested in understanding regulatory mechanisms of gene expression? Are you trying to unravel how and which regulatory elements are involved in this process? Perfect, then you're right here. Today I want to introduce our web server EpiRegio, which is a resource to study regulatory element-gene interactions. My name is Nina Baumgarten, and EpiRegio is a joint project with Dennis Hecker, Shivaranjan Karunanidhi, Florian Schmidt, Markus List, and Marcel Schulz. A current challenge in genomics is to interpret non-coding regions and their role in transcriptional regulation of possible faraway target genes. And these regulatory elements can enhance or repress the expression of their associated genes. And the first question to answer is how to identify these regulatory elements. And the second question which comes in mind is illustrated here with this small cartoon, where you can see one gene and several regulatory elements. And the question now is which of them are associated to the gene of interest. And identifying these target genes of the regulatory elements is an essential step to understand the impact on the gene expression. To predict regulatory elements, we used a tool called StitchIt, which was mainly developed by Florian Schmidt from our group and Alexander Marx, which is a member of the group from Gilles Frecken from University of Saarbrücken. StitchIt is an algorithm to identify regulatory elements and simultaneously their target genes, and therefore it interprets epigenetic signal variation of many samples in relation to changes in gene expression. It's based on paired DNA-seq and RNA-seq data. Here in this figure you can see the idea illustrated on 3 samples, and for each sample the DNA signal and the gene expression is shown. In the first and the third sample the gene is highly expressed, indicated by this high gene expression value, and in the second sample the gene is basically not expressed, shown by this low gene expression value. And if you look now at the DNA signal, you observe several peaks which could be relevant for the expression of the target gene. The first one here occurs in all 3 samples, independent of the expression value. So what does this mean? Maybe the peak is important for the initialization of the transcription, but it's not relevant for the amount of expression itself. For the second peak, we observe an association. If the gene is expressed, we see a peak, and if the gene is not expressed, we do not see a peak. For the last peak here, it's exactly the same situation, just the other way around. We observe a peak if the gene is not expressed, and we do not observe a peak if the gene is expressed. And these 2 areas here shown in green are recognized as important for the amount of expression and therefore predicted as regulatory elements from StitchIt. To solve this problem, StitchIt uses a segmentation-based method followed by a 2-level machine learning approach. The data hosted by EpiRegio was generated by applying StitchIt to 110 samples from the RoadMap Consortium and 56 samples from the Blueprint Consortium. And for the data provided by EpiRegio, we considered a window of 100,000 base pairs upstream of the gene's transcription start site, the entire gene body, and a window of 100,000 base pairs downstream of the gene's transcription terminating site. So we even take distant regulatory elements into account. In total, EpiRegio contains more than 2 million regulatory elements, which are associated to more than 35,000 protein coding and non-protein coding genes for 64 tissues and cell types. And now I want to show you our web server in a live demo. This is how the EpiRegio website actually looks like. We provide here 3 kinds of queries: the gene query, the region query, and the realm query. And today I want to introduce the gene query. And for the gene query, users can enter gene symbols which they're interested in, and then we will return all the realms associated to that gene. So let's try with EQ1, for instance. Instead of entering here the symbols one by one, you can also upload a file which contains all the genes you're interested in. And here additionally you can enter a cell type or tissue, and we now want to try it with a heart, and then you can query the database. And this is the result. Actually each query resides in the same interactive table, and I just want to draw your attention on 2 entries here. The first one is the model score, which is an indicator on how much of the target gene's expression can be explained by this realm in relation to all the other regulatory elements associated to the same gene. And the second one here is the cell type score, which is here called heart score, since we picked heart as tissue. And this is the normalized quantity which represents the relative contribution of a regulatory element to its gene expression in a cell type specific manner. Next, I want to illustrate how EpiRegio can be used to identify transcription factor target genes, and to do so, ChIP-seq peaks alpha TF of interest can be uploaded to EpiRegio's region query, and then EpiRegio searches for regulatory elements overlapping a given percentage with these peaks, and after a functional enrichment analysis based on the genes associated to the overlapping ramps can be done. And with that, EpiRegio can help to identify pathways that are affected by the TF of interest. Additionally, EpiRegio can be used to identify enriched transcription factor of differentially expressed genes. And therefore, we upload the differentially expressed genes to EpiRegio's gene query. And then, based on the associated regulatory elements, we perform the motif enrichment analysis. And to do so, we use a tool called PASTA, which allows us to include the cell type score within the prediction. And by that, we identify key transcription factors of enriched in the regulatory elements in a cell type specific manner. To sum up, I introduced EpiRegio, our new web server, and EpiRegio not only allows the analysis of genes and their associated regulatory elements, it also provides the activity of the regulatory elements and the estimated cell type specific contribution to its target gene expression. Further, it is possible to explore genomic regions for their regulatory potential using the region query of EpiRegio, and you can investigate overlapping regulatory elements by using the concept of cluster forms. And additionally, it's programmatically accessible using our REST API, and the website itself is freely available. For a more detailed description of our work, please have a look at the preprint of CIGIT and also of the EpiRegio paper. With that, I want to thank you for your attention. Check out our website and feel free to contact us.